This episode of Beauty and the Surgeon podcast is brought to you by Let's Get Checked. Let's Get Checked is an online company dedicated to making professional health testing easy and accessible. Let's Get Checked offers fast, affordable, completely confidential at-home health tests from a range of tests from STDs, male and female hormones, and even COVID-19. Listeners of our podcast who are new to Let's Get Checked get 20% off by using our URL, trylgc.com/beauty, and be sure to use the code BEAUTY20 at checkout. That's trylgc.com, trylgc.com/beauty, BEAUTY, and be sure to use the code BEAUTY20, BEAUTY20 at checkout. Do it. Getting tested is important. It's the responsible thing to do, and you know how important I think it is to make sure you understand your labs. Welcome, everyone, to Beauty and the Surgeon podcast. Head bobbing. Amy, I'm a nutritional therapy practitioner. Like, seriously, what are you doing?
Well, I'm thinking about paying my tax payment.
Oh, well, I'm joined today, as always, by the head bobbing.
I just want to avoid, can we just make this episode go on for a couple weeks?
So the tax day just goes away? Well, it's already happened. It's here. It's the 15th.
Oh,
it's
it is
happening.
the 15th,
yes. The board-certified plastic and reconstruction surgeon, Dr. Martin. How you doing?
How you doing, Amy?
I'm doing good. I mean, literally, this is what I'm seeing out of the corner of my eye.
Yeah.
Mm-hmm.
That's how I'm feeling today.
That's it. You're bobbing. I like it. Head bobbing.
So what are we doing?
Well, so today we were talking about research we're into. I called it 4.0 and I had a question mark when I sent these to Sarah because I'm like, is it 4.0? 4.0? I think it is. So if you would like to see the slides, there's not super engaging slides, but there are a couple of cool pictures that are really neat to see, and they do kind of help explain some of this research that we're into. You can find those on our on our YouTube channel at Jason Martin MD.
Yep.
It throws me off when you stare at me with those crystal clear eyes.
Again with the eyes.
Not used
Again
to
with
it.
the eyes.
It's unsettling.
Well, your makeup is nice.
Oh, thanks.
It's like a nice shade.
Well, I'm trying to make it so that Nils doesn't have to try and photo edit out the shining head that I am. So
Amy and I are really ugly, and Nils just edits us to.
That's right. Well, we talked about that earlier. We're just going to become avatars pretty soon.
Oh yeah.
We'll just be a voice.
We did. We talked about anime avatars with very large eyes.
Yeah, I think that's doable.
Right.
Yeah. So we're going to talk about research we're into. And I love these podcasts because you and I very often have conversations where we talk about research we're into.
Now, do our listeners love these podcasts? That's the real question.
Well, I guess we'll find out. So if you like these podcasts where we talk about research, leave us some comments, some feedback, some ratings.
Even if you hate it, leave it.
Right. If you hate research, tell us that too. But we're into it, so we're probably going to keep doing them.
Your research podcast really makes me angry.
I mean, that might be the case.
It might be the case.
Yeah, if it does, I'm sorry. Your research article is first. I'm gonna let you control the slides, maybe.
No, you go ahead.
It makes me nervous. All right, so let's, without further ado, get into Dr. Martin's research article.
So just FYI, this is not the 15th, and we're recording this on a Saturday. And the article that Amy conveniently chose for me, which I'm glad she did, it makes my brain hurt. Okay, so normally on a Saturday, I'm chilling out. What do we do on Saturdays, Amy?
I usually work.
Yeah, we work. That's true.
I don't know what you do on Saturdays, but I'm usually
Yeah, you're right. I do the same thing too. Anyways, theoretically speaking, I'm relaxing, maybe having a cup of coffee, playing with my puppy. But so this was a very difficult article to read. But
as much research is.
Right. There's a couple ways you judge articles. Number one, how many pages it is. This article was 18 pages. Okay, that's 9 too many. Secondly, if it's from China, okay, which they're amazing scientists there, don't get me wrong. So this is not like being against Chinese scientists. You know that the paper is going to be extremely technical, right? I'm sure the same is true if you get a paper from Switzerland or, these places where people are very methodical. Like the good thing about American papers is they'll give you the synopsis. They'll give you some flavor in there.
There's the abstract right there.
I mean, this paper was very difficult to read.
I'm going to interject quickly. So it was funny when I was doing the slides for this podcast, my brain was a little bit fried. And I said to Catherine, who is our front desk assistant, you read through these research articles, sometimes you think this is someone's life's work. And really people in their circle might read it and no one else. And it might not ever actually mean anything in their lifetime.
Which is crazy.
But that's the majority of research.
That's like all of research.
It's rare that someone
and you read a
hits
paper
on
like this,
something.
and you're so amazed at the thought process, the intelligence, the time they spent. Yeah. So we'll get into it. Anyways, it was in the Journal of Neuroinflammation. The title was Sirtuin 3 protects against anesthesia and surgery-induced cognitive decline in aged mice by suppressing hippocampal neuroinflammation. So in normal person speak, what that means is there's this sirtuin peptide, this protein, specifically in the brain that can help protect you as a patient or a mice as a patient.
A mouse.
We're
a
assuming
mice,
that it affects humans too. And
a mammalian.
Yeah, there's a common thing you see in aged both people and mice. You give them general anesthesia with an inhalational agent. Afterwards, they have a lot of cognitive problems, okay, memory and learning. So anyone listening to this podcast has, who's of middle or higher age probably has thought about this because you hear about this. These friends or someone or you read an article where someone's had general anesthesia and they're not the same afterwards, right? And so it's really interesting. And the cool thing about this article, and this is why Amy's so great, it combines the stuff that I'm really into, longevity and sirtuin pathways, with surgery, which I do also, and the effects of anesthesia after surgery, specifically on people that are older.
Yeah, that was one question I did have, and I'm sorry if you're going to touch on this, is what did they define as an elderly person?
They didn't.
Right, because if you're over 34,
because when you're in China, you don't have to worry about that.
Right,
you just tell exactly what you did and exactly what you found. And
elderly.
There's no discussion. So they didn't. I'm sure they described what the elderly mice were, but we're equating a mice study to humans, which is always a big problem. But that's the best thing we have.
Well, no, but that's my point. We see these complications in elderly humans.
Yeah, it's over 65.
Over 65.
Yeah, and so what I found was after non-cardiac surgery, for patients over 65 years of age, 65% experience delirium and 10% experience long-term cognitive dysfunction.
Wow.
And that is from JAMA. And I know these days no one believes in science anymore, but JAMA is pretty important. So if you're over 65, you're going to get general anesthesia, which, again, they didn't define in that statement, they didn't define what kind of anesthesia they got. And we'll talk about that. Then you need to think about this. So that's what this study actually looked at, and it's extremely interesting. Let's go on to the next slide.
Well, before you get too far down this line,
okay.
I think it's important that we talk about sirtuins.
Yeah.
I would honestly like to have a podcast that's just titled "Let's Talk About Sirtuins," but that could be an entire show, not just like a.
There would
episode.
be 4 people listening to that podcast. It would be Nils, you and I.
Only because he has to. Yeah. Yep. So
and maybe Sarah.
Sirtuins are fascinating. And Dr. Martin, you mentioned that sirtuins are proteins. Sirtuins in mammals, there are 7 different sirtuins that we are aware of. And I say that because, 100 years ago, no one had any idea that sirtuins existed.
Right.
But where the sirtuin is located in the cell is different. So the sirtuins 1 through 6, primarily sirtuin 2 is in the cytoplasm, which is, I wanted to be able to get your picture of the cell that you drew.
She's talking about Jason Martin, MD. You got to look on her YouTube channel.
But you drew one for what podcast episode was that? Fasting. The one where we talk about fasting. Dr. Martin drew this really cool picture of the cell. And I was like, I wish I could get that and do the sirtuins in it. But sirtuin 2 is primarily in the cytoplasm. Sirtuins 6, 7, and 3 are primarily in the nucleus. And then sirtuins 4 and 5 are in the mitochondria, right? I think
you're kind of messing up a little bit. Yeah.
Yeah. 6, 7, and 3. But they go back and forth, and this kind of shows it that Sirt 3 can go from the mitochondria to the nucleus. Sirt 1 is actually primarily in the nucleus with Sirt 6, but it needs to work in conjunction with some of the other sirtuins. So
yeah.
Sirtuins are tiny little things in something that is already super tiny. You think of the size of a nucleus, and 3 of these sirtuins are inside that.
Totally.
And sirtuin 6 alone has 240 or 250
subtypes.
No, amino acids just inside of it.
Okay.
So,
yeah, and then no.
Sorry.
No, I think it's go back to so interesting. So, eukaryotes, plants have sirtuins, right? So that's why when you drink wine, they actually have sirtuins in there, and that's why it's supposedly healthy for you because you're ingesting sirtuins. So
it has resveratrol, which activates sirtuin.
Right. I'm sorry. Yeah, it has resveratrol, which supposedly is an activator of sirtuins. Now, we talked about before in multiple episodes, you either have the mTOR pathway or you have this pathway. So mTOR pathway, you have a lot of energy, a lot of eating, a lot of calories on board. You're going to build stuff. And you have this pathway over here where you have depletion. You don't have calories. You don't have food. And you're going to clean house. We talked about something called autophagy. We talked about the AMP cycle. And what's the other ones that you like, Amy? I don't know. No one's going to even care.
Oh,
okay.
okay.
Yeah. You know what I'm talking.
Go listen to our podcast about fasting.
Yeah, okay. So basically, there's two options. Either you have enough energy, you're going to build stuff, or you don't have enough energy, you're going to clean house and cause autophagy or, lean on autophagy and, all these other pathways and stuff like that. So with aging, this is what's so interesting. Specifically with aging in the brain, you get cognitive decline. Now, Amy, have I gotten there yet?
No.
Okay, good. So
ask me again. No,
right. We're not really sure why that happens, but we know there's a couple things going on. Repeated inflammation is definitely one of them, right?
Chronic inflammation.
Yeah.
Yep. Silent inflammation.
And number 2, energy production. One of the things we've talked about before, and Amy's said to me multiple times, taking creatine in the morning will increase the ATP production. ATP is the energy currency.
Specifically in the brain.
In the brain. So there's things going on as you age. You're not making as much energy, and energy comes from the mitochondria. And you're getting a lot of inflammation. Inflammation actually does come from the mitochondria too. Sirtuin 3 is actually in the mitochondria. And it actually helps control or decrease inflammation. And it actually helps you increase energy production in times of depletion. So this Sirtuin 3, they've postulated that is really important to longevity and to maintenance of your brain. We've all seen those apps about neuroplasticity. Where the old you do the words or crossword puzzles or stuff like that. They talked about, kind of
not fair to somebody who's dyslexic.
I know. Okay. Keeping your brain active.
You do the words. No.
Well, Sirtuin 3 has been shown to actually increase neuroplasticity to help you lay down new nerves, even at an older age. So the Sirtuin 3, through a lot of different research, has been shown to be really positive for your brain. And again, Sirtuin 3 is made at times of depletion. So calorie restriction, fasting, which we just had a great episode on that, and exercise.
Well, it's not made so much as it's upregulated.
Upregulated, yeah. Thank you. Whatever. Or you can take pharmacological things that would upregulate it, or I guess they would upregulate it. Okay, resveratrol, I take fisetin. And then it gets even more complicated from there. You need NAD, which if you listen to any longevity podcast, they talk about NAD. It's an NAD specific process that's necessary. So NAD needs to be there, and that definitely goes down with age. So that's where we are with Sirtuins. So these really smart scientists decided they're going to take mice and they're going to look and see truly what Sirtuin 3 does in relationship to anesthesia-induced cognitive problems. So why does that matter, Amy? Learning and memory comes from what part of the brain?
Hippocampus.
It's not your medulla oblongata.
No, it is not.
It's your hippocampus, okay? And in that area, there's an area called CA1. This is very important. You need to memorize this. Okay. No, you don't. Anyways, they studied that area in mice, which I'm sure is equivalent to humans. And they wanted to see if they could increase their Sirt3 concentration in those areas, if there would be a measurable outcome in cognition and learning. The second part of this you need to know, they've come to find out that these anesthesia-induced problems are multifactorial. Decreased energy production after surgery, increased oxidative stress, and decreased neuroplasticity. And they think that's because the Sirtuin 3 is downregulated after anesthesia. So some process with, getting these inhalational agents, which is basically they used isoflurane in this study. And so this is what they did. This is crazy. I can't believe I'm even saying this, okay? So they took a control group of mice, and I think they may have given them local anesthesia, and they did basically an osteotomy in their tibial bone. So they cut the bone. And then they gave another set of mice an osteotomy in the tibial bones, in the leg bones, but gave them isoflurane or general anesthesia agent. And then they looked at their baseline ability to learn, both with watching them in their environment with locomotion and also with negative, I don't even get into what they did, negative reinforcement stuff, like with shocking them. So they would, see how quickly they would learn or when they would freeze. And the freezing part shows lack of cognition. And they did that before and afterwards, day 1, day 3, and day 7. Now, this is where it gets crazy. Then they take an adenovirus, a viral vector, and they inject it in their hippocampus. How do they do this? They didn't tell me.
I've seriously been talking about like microsurgery.
Right. And they inject it into their brain, and the virus it gets into their brain and produces Sirtuin 3, right? And they showed that the ones that had the injection with the virus showed a decrease in POCD or post anesthesia, whatever it's called. The decreased incidence of cognition problems after anesthesia, and that's definitive, and that was statistically significant. So it was crazy. Now, they only studied this for 3 days, okay, or for 7 days. They studied it in mice, and they didn't, the way they study mice is they literally put them in a contained area, and they watch their movement with a camera, and they analyze it, I'm sure, with AI stuff. And then they did some of this like fear feedback stuff with shocking and sounds, and saw like how they reacted and how much they freezed.
Well, yeah, and a lot of and even humans, like if a loud noise will cause a person to freeze or an animal to freeze, because then you have to decide like, fight or flight,
right.
Like what was that noise? Should I run? Should I fight? Yeah.
Right.
Yeah.
And then afterwards they did all these studies. Okay. So, I mean, these poor mice, because they're not with us anymore. Rest in peace, mice. So they looked at, they did a Western blot, okay, to assess the protein concentrations, which is basically the Sirtuin concentration. They did immunofluorescence to assess microglial morphology.
Oh, I love the microglia.
And which is, we've talked about that before. That's like the kind of immune system around the brain that controls everything.
Lymphatic system.
Yeah, lymphatic system. They did ELISA analysis, ELISA, which I learned about at medical school, to look at like TNF and IL-1, all these kind of cytokines that cause inflammation. We've learned all about those with COVID. They did spectrometry to look at superoxide dismutase. So this oxidative stress that happens with anesthesia or with aging, the Sirtuin 3 actually has forces the formation of superoxide dismutase, which helps. It's an antioxidant. They looked at the Golgi-Cox staining to see synaptic structural plasticity, et cetera, on and on and on. So these people did like 1,200 studies into one, and that's why it was 18 pages long. But it was pretty remarkable. Everything showed, decreased inflammation, increased energy production, increased plasticity for people that had the viral injection of basically in formation of increased levels of Sirtuin 3.
Did they only do that after the surgery, or did they do it before and after?
Before.
But not after. So like, they didn't do it after to see if they could reverse the effect of the cognitive decline?
Yeah.
No.
Yeah. And what they're finding, and this is where it gets so interesting, and I don't even think Amy knew that this is where this article was going, but microglial activation leads to neuroinflammation.
Yeah, you don't think I love the lymphatic system enough to have found this?
So alterations in microglial activation and neuroinflammation have been associated with synaptic plasticity dysfunction and in neurodegenerative diseases like Parkinson's
and Alzheimer's.
So the microglial system is about inflammation of the brain, and they're looking at Alzheimer's disease, Parkinson's disease, ALS, like all these things that lead to these degenerative neural disorder, Huntington's disease. And they're finding that basically it's related to this inflammatory process, that actually this Sirtuin protein can actually improve. The question is, if you increase Sirtuin 3 in people's brains with these problems, will they get better? Imagine in the future, 10 or 20 years from now, they're injecting viral, vectors. How do you? I know this gets so confusing.
Well, the adenovirus is just the carrier. It's
the carrier, the virus, injecting a virus in your brain
with the other
that implants some sort of
substrate
genomic material into your RNA or DNA, wherever it comes from, and then you make this peptide called Sirtuin 3, and all of a sudden it decreases inflammation, increases neuroplasticity or your ability to make nerves, and then decreases energy production in your brain. All of a sudden you're cured of these problems. By the way, my dad, when he had a deep brain stimulator with Parkinson's disease, he was entered into a study and actually didn't do it for a lot of different reasons, but the study was to do nerve grafts at the same time of the placement of the deep brain stimulator to put these grafts in.
Where would they get the graft from?
They got it from the foot, from the leg.
Okay, so they take it from the leg and put it into the brain.
And I don't know the specifics of the study, but I think it was healthy components of nerves to hopefully stimulate neuroplasticity and I'm assuming to form almost like stem cells to form new.
Yeah, like to make interesting.
Yeah. So
like the little dendrites in there, like
right.
from a foot.
So you can imagine if you're putting a deep brain stimulator in for Parkinson's disease in an area where you're having high inflammation, destruction, no neuroplasticity, no new nerves, and they're not functioning correctly, and you're putting the deep brain stimulator in there, and at the same time you inject a virus in the same area that increases Sirtuin 3 peptides or whatever other peptides that are helpful, that could have a drastic effect on these people's prognosis and how long they can live with the disease. And that's a lot of these chronic diseases like Parkinson's disease and Alzheimer's, which it's so funny to talk about this stuff because it's so common now. Mm-hmm. Like everyone knows someone with Alzheimer's that it actually all of us should be thinking about this. They may end up being like HIV where you can just live with it, right? Where we have these palliative, almost like things that help make you normal, but you're not going to cure the problem. Right.
Right. I think that's like MS is maybe a good example of that. Like it is much more. It's much less of a death sentence than it used to be.
Right.
Like when I was a kid, I was in school with a boy. His name was James, and he had MS, and we'd gone to school together since kindergarten. But by the time we were in he started out with a walker and then a cane, and by second grade, he was in a full-on wheelchair, or second or third grade. And I don't know what happened to him after. It was one of those things he just never came back from summer.
Yeah.
So but now people live lives with MS. But they have massive drug cocktails that kind of keep their symptoms suppressed.
Yeah.
Like the expression of the MS is suppressed.
Yeah.
So maybe the expression of the Parkinson's could be suppressed.
Right. I mean, the etiology of Parkinson's and Alzheimer's disease, we don't know. But even when it relates to surgery, why is it that anesthesia causes inflammation in your brain?
Right. Well, and why wouldn't your body be able to fight it? I guess that's why I asked if they did that same injection after to see if they could reverse the effect that the anesthesia had.
Yeah, they did not.
That would be so interesting to see.
Yeah, and they didn't try different inhalational agents, different anesthesia agents. Well, that's another issue.
What the compounding effect would be of pain of opioids after that as well.
Right. So that's where it gets complicated because most of our surgeries, we use propofol, which is a very short half-life. You get it with colonoscopies and things like that.
Yep.
So the likelihood of them causing a postoperative cognitive dysfunction, POCD, is probably very low. Whereas you get in these inhalational agents that are very strong, like isoflurane, you get paralytic, like rocuronium, and all these different types of strong medications. That's where I think you can get this inflammatory process in your brain. And it's probably related to the downregulation of both, Sirtuin 3 and some other factors, NAD, whatever it is. Mm-hmm. And then afterwards you wake up and you're in this delirium or this haze. I went through that with my general surgery procedure, and they probably didn't use strong medications.
And you're young.
And I was young, mm-hmm. which is a little bit concerning. So the Sirtuin levels in young people is about double what it is of 3 is double what it is in adults and over 65. So that changes over time. Your NAD levels in young people is about double what it is when you're 65 or older. So all these things go down, these neuroprotective compounds go down, and these insults, you can't recover from them as fast. Now, what can you do? Okay, let's talk about this real article, possible human implications. Well, you can't do much. If you need surgery, you need surgery. Clearly, also, if you're having elective surgery like cosmetic surgery, just make sure they use the least amount of anesthesia they need. This is why we push people to do in-office awake procedures, because there's no anesthesia for those procedures. Secondly, there's a lot of risk factors associated with this. Amy and I were talking about it: hypertension, sleep apnea, diabetes.
Which makes sense because, in the nutritional therapy world, we call Alzheimer's type 3 diabetes.
Yeah, that's good. Drinking, depression, nutritional status, social support.
Causes inflammation anyway.
So all these things that are problems, you need, if you're fortunate to have time before surgery, you need to maximize those. Stop drinking before surgery. If you have sleep disorders, wear your CPAP. Depression.
Get your blood sugar under control.
Yeah, make sure your diabetes is under control. Depression. Nutritional status. Make sure. We have an amazing episode where Amy talks about nutritional support around the time of surgery. So I think it's really important that you do everything you can in your world to make it better. And then you have a conversation with your surgeon about the type of anesthesia they're using and trying to limit that as much as possible.
You know what else I think would be interesting in this study is if, like how someone's life work, I'm not saying they didn't do a good job, but if they took the mouse and put them, because they did the like training with them before the anesthesia, how would a mouse that didn't get that training fare against the ones who did get it? So like even the old mice that went through the training and still got general anesthesia, what would they do with a mouse who didn't go through the training and got the general anesthesia and then tried to train them? Like, how would they function?
Right.
So maybe even things like doing the word, like Wordle or playing word games or whatever to like kind of get your mind like firing really well before surgery and then starting it right away after, like maybe that'll bring you out of it quicker.
Yeah, that's a good point.
I don't know. There's so much. Like I think the human implications are massive with this.
Yeah. And the baseline of all this is we say, okay, we age. Why does our brains change? It's frustrating as you get older. You can't remember as much. Well, it's two things. We talked about inflammation. We talked about energy. Neurons, which is what your brain's made of, basically is a bunch of nerves, take some of the highest amount of energy to function.
Your brain is hungry.
So like besides muscle, when you're really working hard, your brain requires so much energy, and the mitochondria is where this energy production occurs. That's where ATP is made, which is basically like the gasoline or the diesel fuel or the sunlight, or the sunlight converted to energy, where it's made. And you could see why it's so hard after you've had an insult, and it doesn't have to be anesthesia, like a stroke or
head trauma,
a head injury, or a severe medical event. Why it's so hard to recover mentally, because your body's depleted. So maximizing yourself physically before surgery has a lot of benefits, not just living or your heart's healthy or your organs are okay, but your brain can recover quicker. And I can tell you the postoperative delirium, I experienced it. It was terrible.
I was upside down world.
Yeah, I didn't feel right. I was confused. And some of that may have been the narcotics, which I only took one, but I'm a teetotaler, so is Amy. We don't take narcotics, unlike most of Colorado, who seems to enjoy narcotics. But, it's interesting to be in that situation. It makes you really feel, just to have that experience of confusion a little bit, and someone who's very acute, it makes you feel for people with neurodegenerative problems, right?
That's just how they function.
It's almost like you can be empathetic. And the fact that maybe, obviously, Sirtuin or Sirtuin 3 is not going to solve the problems of neurodegenerative problems, but if that's one of many things that you could do.
Right. Like I said, who even knew that existed? When you were in medical school, did they talk about Sirtuins?
No, they didn't talk about it. They barely talked about NAD. That was only in biochemistry.
Yeah.
So,
and they probably didn't even really understand like why it was necessary.
No. What is it? David Sinclair just came out with his papers like in 2015.
Yeah, this is all new.
Yeah.
New and emerging for sure.
Yeah. So if you want to do something that's positive before surgery, the most important thing is to maximize your health and your mental well-being and your social support. Also, if you want to do like super special things, we have no idea if that's going to make any difference on postoperative cognitive dysfunction. You could take things that stimulate the production of sirtuins.
Or even like NMN. Like I wouldn't. There's really no contraindication for MNN before surgery.
NMN or NAD before surgery. You could take resveratrol. You could take fisetin or all these other supplements that increase sirtuin levels. I think all these things, you could take creatine actually too.
Yeah, creatine I would highly recommend.
So these are things you could do real time now that actually benefit you. And I would do that even if I wasn't over 65.
Yeah.
All right.
Is that it?
I feel like Will Ferrell at the end of Old School when he's at the test. You know what I'm talking about? And he's like, okay. And he takes it, he exhales. He goes, I don't know what that just said. No one's going to get that reference unless you've seen Old School. Yeah.
Yeah. I thought that was super awesome. That was very, very interesting.
I blacked out.
Lots of slides coming up. So any final takeaways?
No, I think it's really interesting. This study was super intricate. Only Chinese scientists could even come up with this idea. It was amazing.
But it could have some really cool human implications.
But if you like longevity and you like brain health, read about Sirtuins. Okay.
And the lymphatic
system, inflammation and stuff like that. You'll find it really fascinating. It's really helpful for you to make, kind of healthy decisions for your future.
Awesome. Well, let's take a quick break to hear a word from our sponsors. Let's get checked. And then we'll come back and talk about my research.
Let's do it.
Dr. Martin, I'm super excited to talk about our sponsor for this podcast, Let's Get Checked. If you've been a listener of our podcast, you know that we have never had a sponsor before, and that's because we're an educational podcast. We're not looking to monetize, and we also would never recommend something that we didn't believe in and or use ourselves.
So basically, you're saying we're very choosy.
We're very choosy when we come to our testing, but Let's Get Checked really aligns with our values. We talk a lot about monitoring your blood values of certain things. We talked recently about vitamin D serum levels. We talk a lot about hormone levels, and it's hard for some people to get to a lab, or you're afraid of the blood draw, or just, you're concerned about confidentiality. Like, you have a lot of concerns and you just don't do it. Let's Get Checked makes it super easy because it's completely confidential. You do it from your home. So there's really no excuse not to do it.
Yeah, and we did it recently. Both Amy and I did it. She looked at her women's health, basically her hormone levels, and I looked at mine on the men's health side with testosterone. It was very simple. You go online. Their online interface is really easy to use and good. We got the testing kit very quickly. And the test itself, actually doing the stick, you do a stick on the end of your finger, is not hard to do. It only took about 3 to 4 minutes. You put it back in a sealed envelope that's confidential, goes back to them. You get your results in 2 to 5 days. It's really that simple. And the best part is it's about empowerment, about you taking control and trying to do things that are positive for your health and life. And this is one way to do it, literally from the comfort of your own home.
Yeah, so they offer a wide range of tests. So they do men's and women's hormone panels. They do a full panel of STD testing. They also are now offering a COVID-19 test at home. So there are a lot of other just basic blood levels that you can get tested as well. And one of the things that we always say is you can't track what you don't check. So you might not know what's normal for you in the future if you're not checking it now, especially as it relates to hormones. And men's health specifically, I feel like, is a little bit undertreated. I mean, sperm counts are dropping by a large percentage in men these days. Most men struggle at some point in their lifetime with low testosterone.
And those men do not know what the symptoms of low testosterone are. Depression, anxiety, sleep problems, osteopenia or loss of bone, loss of muscle, and something we treat all the time, gynecomastia or male breast growth. These are real problems that can be improved or treated with testosterone. And these men do not know their levels. It's so easy to get checked. And this is why we're so excited about the sponsor.
Yeah. And Let's Get Checked is a completely CLIA certified lab, the CLIA lab. So they're a well-established, completely safe lab to use. You do get contacted after, so you get your results. I got a text at 4:30 in the morning, which I loved, that my results were ready.
And we're both up at 4:30,
yep,
so that was good.
Super excited. And you have access to not only a nurse, but also a physician. So a physician does review your results, and it gives you a little breakdown of like where you're sitting on those levels. Dr. Martin's had even comparisons of like where 2 of the levels should be in relation to each other, which is a really important thing to note.
Right.
Just being within the normal range is not always enough. So it really gave more information than that. You'd still want to, of course, reach out to your own doctor if you needed some additional assistance with these labs, or you did see something that was concerning. We didn't, thankfully, so that was good.
Yeah. We were both good. This podcast is about empowerment. It's educational. In my private practice, I'm a board certified plastic surgeon. We are all about empowerment, and this is one way to do it. So we're very excited about the sponsorship, and we're looking forward to using it for our patients moving forward.
Yeah. So as a listener of our podcast, if you're also a new time user to Let's Get Checked, which a lot of you probably are, we do have a 20% discount code. So you do need to go to our specific URL, and it is linked in the description box below, but it's going to be TryLGC. So TRYLGC, like Let's Get Checked.com/beauty, BEAUTY. And you'll need to use the code BEAUTY20 at checkout. Beauty again is BEAUTY 220. And you can pick a test, like any test you'd like, put in our code and get your test shipped to you. Getting tested is the right thing to do. It's lets you know where you're at and what's responsible.
All right. Well, let's get tested.
Let's get tested. Let's get checked.
Let's get checked.
All right, we're back. There is no test yet to test Sirtuin levels.
No.
Nope. Maybe that's coming.
Well, you could cut your brain open and do a Western blot.
That'll do it.
In the Chinese study.
It's pretty final analysis, though.
To get your peptide level.
Yeah, maybe not the best way to find out. All right,
I think like a needle in your hippocampus was probably not good.
No, I'm thinking that sounds terrible. Yeah. Sounds like a real bad day.
Yeah.
All right. So I'm excited about my research is talking about Sirtuins as well.
Okay.
Which is why I thought it was important that we kind of broke down what a Sirtuin is at the beginning.
Do you think we could get sponsored by Sirtuin or no?
I mean,
sponsorship.
I don't know that Sirtuin
like has a Sirtuin society
has any money to offer. Maybe just some longevity benefits.
Yeah. Some free supplements.
Yes. So my article is not like yours. It's not like a singular research article. It's also not a meta-analysis. It's a kind of broad overview. So my article is 13 pages long, but the last 3 pages of it are all referencing the articles or the studies that are referenced in the article.
Well, that's good.
So basically it's a conglomeration. And the reason it's not a meta-analysis because it's looking at many different studies that are all about different things and kind of finding the correlations to the Cert6 connection. But that's not, I mean, it's not like you're looking at like one type of study and then able to do a meta-analysis. So it's really just a grouping of lots of research that's been done over the last 10 plus years and kind of trying to figure out what might be going on with a lot of these things. It's very interesting. It's not a super challenging read. I would still say it's a very technical read, as anything in the cellular biology magazine is going to be. It's kind of hard to get through lots.
Yeah. What is the journal?
It came from Cell Press, is where it came from, and the article is called Sirtuin 6: Linking Longevity.
I mean, once that comes out, I read every page front to back.
Right, with genome and epigenome stability, and this became. A lot of hype was kind of made around this last year in 2021 when this kind of came to light, like, oh, the longevity connection, but why? Right? Why does Sirtuin 6 help with longevity? And that's what I was really more interested in, and that's why I found this kind of broad overview of so many research papers in one. It gives you kind of, it's like finding all the little, like the breadcrumbs. Right. So you're picking up all these little parts of all these different studies and finding out why it helps with longevity. Because, Sirtuin 6 is one of the ones that's located in the nucleus, along with Sirt1, and I think it's either 5 or 7. I think it's 7 in the nucleus back there. So what's it doing? Overexpression versus no expression is kind of what they looked at, interestingly. So if your study, they just had one type of mouse. Looking at all these studies, they actually had a whole group of mice that were these knockout mice where they had no Cert6. So they were specifically bred to have no Sirtuin 6. And it depended on what breed of mouse they were, how this affected them. So some genetic, mice were these knockout mice would die almost immediately upon birth. Some would live for up to 10 days. Others, with these knockout mice, would live, some of the females lived for almost 100 days, 300 days, and 10% of the male mice lived for 300 days. So it was very much an oddity, right? So that just makes it even more conundrum-y. So genetic background can make a big difference too. So even if you take it out entirely, it still depends on what genetic version of a mouse you are, how long you're going to live. In rhesus monkeys, if they knock out Cert6 in those, they die immediately.
That's crazy.
They do not live.
Because you think about the idea of sirtuins in general, they're mustering up energy when you don't have any. And the ways you do that is break down fatty acids or fats. You break down proteins, right? You can use that. I mean, all these kind of different, and I'm sure glycogen, that kind of stuff. It's all these and I guess when we talked about my part, we didn't talk about glycogen because they don't really store glycogen in the brain, do they? Mm-mm. Yeah, that would be in the liver. So, you realize what it's doing, right? It's trying to muster up energy just to have normal processes go on. And go ahead.
Well, Cert6 is different than Cert3 because it's not so much about the energy production as it is about the way the DNA is expressing and repairing itself.
Interesting.
Even. So it's in the nucleus versus in the mitochondria. So I don't think it's so much about having the cell have no energy is the reason that people die. I think it's more that they can chromosomally not replicate.
That's interesting.
And that's why they die.
Yeah.
So in male rhesus monkeys would die before birth and females would die immediately after. So very, very odd, right? And this is why, this is why as much as people hate that there's animal research for a reason. Can you imagine if they were doing this in humans and everyone they did this to just died?
Yeah, well, it's not going to happen.
Right, it's not going to happen.
Well, maybe in certain countries, but no.
Right. So let's take a little bit of a look at, I have a lot of notes, and I could have had notes on this, thousands of pages of notes, but I tried to really pick out the best parts of this research and kind of the parts that tied to longevity the most. So the complex etiology of aging is intertwined with multiple overlapping pathological processes. What that means is the reason we age is not just one thing. And we've talked about this so much on this podcast. There's not one thing that causes aging. It's many things, and it's different in different people, and it's different in different parts of the country, and it's different based on your genetic code and how your genes are expressing. It's not even something that could be the same for the same person across their life. Aging can accelerate, aging can slow down, even in one human, which makes it even more challenging to study.
It's, as we say in medicine, it's multifactorial. There's so many different things
yes,
affecting you.
Yeah. One thing we do know that at least in mammals, the overexpression of SIRT6 promotes beta oxidation in the liver, hepatic lactate and glycerol shuttling, meaning your body's able to use glycerin or glucose better, and it clears it out of the liver more efficiently so it doesn't get built up in the liver. Improves the NAD plus and NADH ratio, which means that it's being utilized more efficiently. Stimulates glycerol release from adipose tissue, so it's getting your fat cells essentially to kind of release their energy, which would then mean you would be able to lose weight more efficiently, perhaps. Improves the capacity to produce glucose. We talked about that, like kind of clearing it from the liver. Alleviates endothelial cell dysfunction, which is something that we maybe in the last couple of years have talked a lot more about endothelial issues. Improves neurogenesis.
Which is what we talked about.
Yep. Helps maintain genomic stability in the brain. And we'll talk way more about this. It also decreases neoplasm in some aged mice.
Which is like cancer.
Yeah. Looking specifically at lung cancers. But if you look at these studies, and again, there's so many, some studies showed that it did and some studies did not show that. Also, the difference between male and female mice was pretty marked. So there's a lot of factors at play. There is strong evidence, however, that SIRT6 deficiency shortens lifespan in mammals, right? Some don't even live, some can't even be born if they're knocked out. So increasing or stabilizing SIRT6 could help counteract aging and possibly influence health span through many factors. So this was, as I mentioned, was not one study. So these are a multitude of studies.
Can I say one thing just about Sirtuin in general? If it's in eukaryotes and it's in mammals, that means this is something from the beginning of time.
Right, because it's in yeast.
Yeah. So clearly this is something that evolution thought was important.
Yeah, this is fundamental to life. Without it, mammals die.
So it makes sense that if you took it out, I don't know the differences totally between 1 and 6 and how important one is over the other, but it makes sense that if you take it out, that really bad stuff's going to happen.
Yeah.
Because it's obviously like a foundational part of
how the cell works.
Yeah, how cells work.
Yeah.
But it's so weird that we don't really know much about it.
Well, and we're learning so much more, but we really don't.
Yeah.
Interestingly, this looked at a group of studies that were performed all the way from yeast all the way up to humans. Like there are studies in humans specifically about Sirtuin-6, and that's why I think it's becoming more fascinating because we are starting to have ethical ways of studying Sirt-6 in humans. But it did start, we talked a lot about Walter Longo's work in our podcast about fasting.
I feel like Walter Longo's definitely an actor.
Right. He's definitely not.
Yeah. Starring Walter Longo.
He does have a really fabulous Italian accent though. Yeah. Okay. When you listen to him. I mean, a lot of his research looking at yeast and how his studies were specifically looking at more of autophagy, but sirtuins are all very closely interrelated, as you mentioned, in the autophagy process. So it regulates, Sirt-6 is primarily a regulator of chromatin, which is basically the stuff that you need to make chromosomes. So it's DNA and protein. And because of that, it's uniquely positioned or strategically positioned, as they said, to maintain genomic and epigenomic stability, as well as influence gene expression in multiple physiological and pathological pathways that determine longevity. Because we do know that part of aging for sure is when your cells, what do they call it? The epigenetic drift, isn't that the term for it? Basically, your cells are just slowly getting bad, right? Like things are just going awry. And you kind of think about it, like every time your cells are replicating, we talked about this, remember?
It's kind of like a party. Those parties you've been to that go too late? You kind of get that feeling.
Right, you get the stragglers left.
Where the poop hits the fan at the end of the night, That's kind of like what happens over life with your cells.
Right, like things
like
are just...
the party started awesome. The music was great. You know what I'm saying, Nils?
Then all the cool people left.
All the cool people left, and then the drinking started.
The crotchety person sitting in the corner.
Then there was a fight.
Yeah.
Someone put on bad music.
The police came. Yeah.
Yes.
It's kind of aging.
That's
in a nutshell
true.
For cells.
And some cells live longer than others. And I think that also with your party analogy, like the last hanger-oners, like that are still there, maybe not the best of the party goers.
Yeah.
The good party goers are gone.
They're all gone.
Yeah. But as things replicate, like as your cells, reproduce and divide, which is happening constantly all the time. Like some do it really fast and some are really slow. Like the cells in your brain are kind of the OGs, right? But other cells like in your mouth, those are turning over really rapidly. So you think about that, all this is happening all the time, and there are mistakes that get made along the way. And that's, your body should identify those mistakes. And if a cell is not replicated correctly, it should destroy it or should just stop, essentially, right? Well, as we age, some of these are allowed to kind of get more into the stream of things. Well, so once that cell has replicated incorrectly once, somebody put a fingerprint on your copier or there's a spot there, that spot's going to keep showing up forever unless you destroy it or you clean it, right? So as you get older, the processes that help
Not
your
to
body.
keep giving you analogies, but we're in puppy training right now, and if you don't start off right with this puppy and you let them get on the couch, they will get on the couch forever. It's the same thing as this. You start out really good, and then things start to slip a little bit, and before you know it, you have an unruly dog.
Right. And the more that you allow this to happen, the less your cells the less your tissues are going to look good. So things like skin aging, if that's going awry, like your skin's going to start to lose some of its ability and structure, like subtly. And that's kind of the drift, like
Exactly.
the drift. But what we found is that Sirt6 plays a key role in DNA repair. So not only in the single strand, but also in the double strand breaks. It goes basically, it's able to recruit the repair enzymes.
Mm-hmm.
So the Sirt6 will go and it'll help recruit all these repair
That's so
functions to
interesting.
happen, right? I mean, like, it's so cool. So it's
You
actually
have one of these things that works like in the mitochondria and the other one that works in the nucleus, and they're both doing different things that are like super important to life and health and longevity.
in a totally different way,
Weird.
but all need NAD plus, like you said. So basically what happens is the Sirt6 will recognize that there's a break, and it will directly bind to the damaged DNA
Right.
and recruit the repair factors.
Interesting.
They found that if it's not there, that does not happen. So it also helps facilitate the orderly recruitment of repair enzymes, like meaning it brings things there basically as an upstream regulator. So they also found, this is what I found so fascinating, that just giving the DNA repair enzymes, like, right, this is what you need to repair your DNA. This is like basically bringing the supplies to someone, like, here, this is what you need to fix this. It's not effective. But when you upregulate Sirtuin 6, you don't even need to add more enzymes because it brings in the correct things in the right order. Like,
Right.
it does it in the right way. Like it's bringing the instructions on what to do, essentially. Like, maybe, and again, it's not like there's an instruction manual for this, but that's kind of what I was thinking. Like, okay, well, that makes sense. So it's an upstream regulator. So basically like having the contractor there, or like when we had to podcast without Nils, did it still happen? Yes. We had the pieces.
Was it terrible? Yes.
Yes. Because we didn't have the person who knew what to do.
It was terrible
here.
lighting. It was terrible sound.
Yeah. So Nils is our Sirt6.
I mean, I watched it. It was like watching a train wreck.
It wasn't
You
that
know?
bad.
Yeah, it was.
I don't think it was that
You were
bad. I
good.
was the one who did it. So I don't
Oh,
think
I'm sorry.
that
It was
bad.
very
Yeah,
good. Did
thank
you do it? It was very good.
Yeah. So with
You were amazing.
Yeah. So I think that's super fascinating because, as we know, as DNA, if it breaks down and is not able to repair itself, your body's just going to start getting rid of stuff, right? So it's not going to be very efficient, which is not good. Like, DNA is pretty key to functioning human life. One other thing that it really helps, and this is where the human study comes in, was it's able to fight that oxidative stress. And they did this with live humans. They take tissue samples, right? Because you can do that. You can do a skin biopsy and look at the activity of the fibroblasts after they'd been exposed to UV light. And if there was no Sirt6 or an underexpression of Sirt6, they didn't repair. The fibroblasts didn't do their repair work like they should. I mean, I think talk about anti-aging, like photo-aging is the number one.
Number one.
Yeah, because we're all being assaulted by UV rays all the time and other physical
Yeah.
assaults to our skin. So if you don't have the Sirt6, your fibroblasts, which are the parts, that are helping your skin cells repair,
Yeah, that's what makes collagen.
they're not functioning.
Yeah.
So over time, you think about that too. Okay, well, so then why are we losing collagen all the time? Why are we losing elastin all the time? Sirt6 is going to be there to help all these things keep replicating correctly and more efficiently. Cells that are lacking Sirt6, this is where, the study brought all the things I love out to play, because then it talked about telomeres.
Oh, here we go.
I love telomeres. So telomeres are the caps on the
That's
DNA.
the only reason you picked this article.
Well, the picture of the telomere
It's like the
definitely
shoestring
got me.
cap.
It's
And
the
then we
yep.
talk about, I didn't think people had shoestrings anymore. And you're like, everyone has shoestrings.
Nils has shoestrings on right now.
Nils is also 100. You guys have never seen him, but he's.
Yes, they have. Episode 100. He was
Oh, that's
there.
right. He was there. God damn it.
Yeah, so really cool thing, and this is where I become the angel of death again. When your telomeres are gone, you're basically going to die because your DNA is no longer protected. So, I mean, as much as Sirt6 is helping your DNA like repair itself, it's going to help slow down your use of those telomeres.
Right.
Because every time your DNA replicates, it loses the telomeres. So if it's not replicating correctly and your body's like, no, that's damaged, no, that's damaged, no, that's damaged, you're just wasting all those telomeres on all those bad replications of that DNA strand. So you're preserving your telomerase and your telomeres, or not your telomeres, your telomeres, by making sure that your DNA replicates appropriately so that it's not wasted or so
Right.
it's not broken. So I thought that was super cool. It also, having a lack of Sirt6, there was telomere and chromosomal dysfunction, which makes sense, right? Because it needs to help make the chromosomes. Whereas when there was an overexpression of Sirt6, it improved the integrity of the telomeres, which is very cool because that is a fundamental thing that we know for aging. telomere health and the telomere length is a key indicator of how long you're going to live.
DNA health and ability to replicate without problems. I mean, that's what cancer is, basically.
Right. And not be broken.
So,
yeah.
Besides trauma, cancer is, well, maybe lifestyle, but cancer is like almost a number one killer.
Well, but what came first, the chicken or the egg in cancer?
Yeah.
I would say that's in some ways a lifestyle disease, not for everyone, but in some
cardiac,
cases.
heart problems.
Right. Those also, though, are lifestyle.
But cancer is a huge killer.
Yep.
So.
Yeah. So that's a really broad overview of like 1,000 studies that were all looked at in this article. But just to give you kind of some of the high points of why Sirt-6 might help with the aging, it's not make you age less, but it's helping your body function more appropriately and like preserving its ability to replicate itself correctly, which that's what you want. Like your body replicating exactly the same all the time is going to keep you younger longer. It keep you being a Sloverine for a longer time.
Yeah, Sloverine.
Yes. You got to Sloverine it. So what was the outcome and what are the possible human implications? And this is where this just speaks right to my nutritional therapy mind, because the outcome is that these are emerging things. We don't know, there is no outcome with these studies. I mean, they're really cool. Like we've looked at a lot of neat things. We know for sure that if you knock out Sirt-6 in mice, they die. Definitely die if you're a monkey, but we haven't been able to do these research in humans, but the things we have done in humans have shown that there is definitely the need for Sirt-6 for repair of cells, especially skin cells. We know that exists. I have this really cool picture here because this is showing all of the basically areas in the body where the Sirtuins are needed. And they're all over the body. I mean, you talked about the brain, when you see an overexpression of these Sirtuins in different areas, and that's what actually was really cool in one of the studies looking at Sirt-1 and Sirt-6 are both in the nucleus. Overexpression in one area of the body does not necessarily benefit, or in the whole body versus overexpression in one area of the body.
Right.
How crazy is that?
Yeah,
you could have a lot of Sirt-6 in your cells or your liver cells.
Yeah.
Doesn't help you at all.
Yeah.
Yeah, like you need them in your brain or the Sirt-1. There's so much. And that's where I go back to, above all else, what can you do? Like, what are the real human implications of this? Properly prepared, nutrient-dense whole food diet.
You can tell me about nutrition. I'm going to go get some of this stuff off the shelf. I'm just starting injecting it in my veins.
Right. Like I have one. Like I actually brought one.
Oh, this is the one I use.
Pterosilbine.
Yeah.
So it says right on the bottle, Sirtuin activator.
Hello.
Right. And you're like, fantastic. I'm going to take this Sirtuin activator and change nothing in my life, and I'm going to live longer. Is that reality?
Yeah, when I take it, it is.
Well, but that's the thing. Like adding things in, there are definitely foods that can increase your Sirtuins. There's actually a whole book written by a guy that's called the Sirt Food Diet, that talks about how to increase Sirtuins. And that's real, right? There are Sirtuin activating foods, green tea, dark chocolate, apples, citrus fruits, parsley, turmeric, kale,
So
blueberries.
I read an article one time that said Ben & Jerry's, specifically Tonight Show, increases, the Jimmy Fallon increases Sirtuin levels. Is that correct, Amy?
I mean, it's not on my list. Red wine.
And bourbon.
And bourbon.
Yeah.
Actually, that might
Mixed
make the
together.
list.
Okay.
So what do people do? They see these, great, like foods that increase Sirtuins. Red wine. Well, why? Because the resveratrol. Well, I'll just start taking resveratrol. Well, resveratrol works specifically on the Sirtuin-1 pathway, which might not actually have the longevity benefits in humans that it had in, let's say, yeast or in mice or rhesus monkeys. Looking at supplementation, nutrients in isolation, or even these are non-nutritive things, right? Like you take exogenous Sirt-6, it's not coming from a food source. It's coming from a made-up thing. Like this is a chemical substance essentially at that point, which we are all made of chemicals. So chemicals are not bad, and that's not what I'm getting at. Food is chemicals, right? It all is. But you don't know the effects. There's also a high risk of developing rampant tumors that they find with overactivation of certain Sirtuin pathways. Because why? Well, it causes things to replicate. Look at your mitochondrial function with the Sirtuins. Like if you rapidly start giving energy to a cell that's not good, you could have a massive tumor, right? That's why that when they would give this, again, in animals, like when you see that there can be a real downside of some of these things, when you think like, more is better, I'm just going to inject myself with Sirt-6 and live forever.
You mean I shouldn't do that?
I don't think so.
Okay.
Or take every supplement under the sun. Now, do I think that supplementation has its place? Absolutely.
Well, Joe Rogan injects NAD. I mean, shouldn't we do that?
Yeah, I mean, NAD, you take liquid NAD, you take NMN in the liquid form.
I'm
so
just joking. I'm totally joking. I mean, and also like you have no idea like what how these things are absorbed, or if you inject it intravascularly, if these are even getting to cells.
Right. Is it even getting absorbed?
Is it transporting into the cell itself? I mean,
should you take NR? Should you take MNN?
Yeah.
Like,
we have no idea what's going on right now.
we don't. But one thing we do know is that food is powerful. And if you look at blue zones, that's what everybody likes to go back to is blue zones. Not yet have I seen a single study on a human population that was taking a supplement and that's why they lived longer.
Right.
They were eating a healthy, well-balanced
Like
diet.
Mediterranean diet with like fruits and vegetables and,
Olive oil.
and no pressed seed oil. I mean, I've heard this. I know the spiel.
Right. These are things, though, that we know. So, like,
Yeah.
if you look at the longest...
But you're
life-
right. You're right. I mean, that's not just their genetic differences. It's the fact that their lifestyle, which has contributed to their nutrition and activity level too.
Right. Their
and
lifestyle
caloric-
and their diet.
and their caloric intake. But it's good. I mean, the downstream effect of that probably has some effect on sirtuins too.
It 100% does. I mean,
So
that
if you
we
want to
know
know
for sure.
why, if you want to know why this lifestyle, fasting, exercise, all these things increase sirtuins, that's the downstream effect.
Correct.
Yeah. Interesting.
Yeah. So taking, I always caution people, like there are supplements that both Dr. Martin and I take. There are supplements that I recommend to people before surgery. There are supplements that are really good, but relying on a supplement when all else in your lifestyle, maybe you have a great diet, but maybe you're exposed to radiation. Maybe, you have other factors.
Like stress.
Stress is a huge one.
Yeah.
ROS. I think of the principle.
Sleep.
Yeah. Also huge.
I feel like this is like summing up all of our podcasts that weren't related to plastic surgery. I mean, everything we're talking about.
It is. And, there's so much is happening to our cells that we don't even understand. But a lot of what's happening to them is coming from external factors, stress, bad diet, exposure to toxic chemicals. I mean, I think, if you live in one of those places where they spray for mosquitoes, I mean, yeah, you're not going to die of West Nile virus, but what are you breathing in? What's being just absorbed through your skin by touching it as I knock my tea over?
EMFs.
My green tea.
EMFs.
Yes, I was going to bring
Plastic,
up EMF exposure.
Lead exposure in the groundwater.
Yeah.
I was reading about that. Like there's like crystallized lead and like,
Mm-hmm.
dissolved lead, and just having a filter is not going to get rid of that. You have to have a reverse osmotic filter to get rid of the lead. And no one's going to get that because it's too expensive.
They're expensive, but they're whole house systems. But even if you were drinking water was reverse osmosis, and then you're standing in the shower and you're getting that lead water all over you, you think your skin is not absorbing that? Absolutely is. So there's a lot of toxic exposure that we are all exposed to on a daily basis. So to think that one supplement could override all of that, or that there was one thing that was appropriate for someone who maybe just had chemotherapy, might have very different needs than someone who, lives on a farm.
Yep.
And is exposed to a ton of pesticides, like very different needs, very different amounts of support. And that's why the one thing you can always stand on above all else that's not going to hurt you is eating a properly prepared, that is key, nutrient-dense whole food diet. That is absolutely never going to lead you wrong. So those are my final takeaways. Do you have any questions about my conglomerate of studies?
No, I love it. I love it. I love the fact that somehow we keep coming back to Sirtuin and it's getting more and more popular.
Well, it is because we know more and more about it. I mean, like I said, 3 full pages of my article were just referencing the articles and not referencing like any more than like just like the name. I mean, like it's pages and pages of what they were trying to look at.
And I think the fasting episode is really important because you and I have both concluded that we also need to have building phases, right? Every time you have a building phase where mTOR is activated, you're really not stimulating sirtuin production.
Actually, that's not true. Leucine triggers mTOR and SIRT6 production.
I was going to get to that. Okay. My point is that in general, if we think about this on a macro scale, but there's going to be ways with nutrition, with supplements, with intermittent fasting, with fasting in general, that you can keep the sirtuin levels up, right? And also build at the same time. You can go back and forth from side to side. Building and
you
taking
are all
away.
the time. That your body is doing that all the time,
but
no
like
matter
as
what.
a lifestyle. I mean, like incorporating that in your lifestyle. I believe that. We're not there yet. I think that people always think there's one magic cure. If you do intermittent fasting, you solve the problems. Well, I mean, you can intermittent fast and have super amounts of stress on your body, which can decrease sirtuin levels.
Intermittent fasting is not appropriate for a lot of the population either.
So
that's just a good example of that is the mouse models in this. You could have 3 different mice, you knock out their SIRT6, but they all are genetically different, like from the different background. Me, you, and Nils, could all have a completely different effect just because of our genetic
I feel
building.
like Nils as a Viking is going to live to like 1,000.
Well, right. You can knock out all of his SIRT2 and
I feel like
he'd
every
still be
Viking
fine.
was 1,000 years old
Yes.
in Valhalla.
That's why. Yeah. So there
and I'm
is
from Kentucky, which means I'm going to die when I'm like, tomorrow.
Yeah, everybody wants to believe that there is one thing, but I mean, that's why I go back to eating a nutrient-dense, properly prepared whole foods diet, because regardless of what your lifestyle factors are, like, yeah, you can eat in a compressed eating window as long as you're eating healthy food. You could eat all day and eat healthy food, and that is still a good way to eat.
Yeah.
Like there is no like dictatorship of diet. There just isn't. And there never will be, because we are not the same person.
Preach.
Yep. Hormones. I mean, we didn't even get into the hormone factors.
No, we didn't even
Like,
talk about
so
we didn't talk
much.
about hormones.
Yeah. So there's a lot. Yeah. You got anything else?
My brain's numb.
I know.
I feel like I just did a facelift.
There's a lot. Like I'm serious. I was so
I would
fascinated
love to see
by
the
this
attrition
research.
during the whole video, like how many people are left at the end. And that's where, again, I think we should do that Marvel thing. We should have a little snippet at the end that's like amazing, but then people would just fast forward. That's what my son always does. He just fast forwards to the end part. Yeah.
He just wants to see
What's going on.
Yep. Yeah. Well, if
All
you
right.
have made it to the end, we really appreciate you guys staying around for this. We love research, obviously. So even if everybody hates it, we're probably still going to continue to do
Oh, we
these
will continue
because
to do
they're
it.
fun and they are
And you will
educational.
like it.
Well, it's hard. I mean, even knowing what they're talking about in these things. I mean, like
If you don't think Sirtuins has anything to do with plastic surgery, you're like,
cell biology does.
right. The fact that we age, the fact that we have wrinkles and our skin thins and all the problems we have after pregnancy and the way that stress accumulates in our body and leads to body changes and deposition of fat and inflammation. You don't think that is anything to do with us treating aesthetic problems, and you're crazy. Like this is literally the reason why we end up being the way we are and why we need cosmetic surgery.
Or want it. That's a brain issue.
We need it. Everyone needs it.
Or wants it.
Yeah.
Yeah.
If you own a business related to cosmetic surgery, I think everyone needs
Everyone
it.
needs it. Just listen to our last episode about Botox and just start getting lots of Botox at 20. So if you're interested in finding us on social media, you can find all of our handles here.
Right down here.
Our phone number, if you'd like to leave us a voicemail, which I very much love, or send me a text, is 303-630-9038. It really does help the algorithm if you give us a like or share us with your friends. That makes a big difference and help getting our podcast out to more people who might also find it interesting.
If you're part of the Sirtuin Club, show it to your friends. When you guys talk about Sirtuins every week at the coffee shop.
Oh, I'm going to get bumper stickers made to say I heart Sirtuins. Yeah. All right. Thank you, everyone, for
That's our
listening.
Podcast jokes.
We'll see you here next time.
See ya.
Don't forget to check out our sponsor, Let's Get Checked. Let's Get Checked offers fast, affordable, and completely confidential health testing for everything from STDs, male and female hormones, and even COVID-19, right from the comfort of your own home. And remember, new customers and listeners of our podcast get 20% off by using our URL, trylgc.com/beauty, and be sure to use the code BEAUTY20 at checkout. That's trylgc.com/beauty and use the code BEAUTY20, beauty, BEATUY 20 at checkout. Get checked. It's the right thing to do.